Before overturning evolutionary paradigms, biologist Sean B. Carroll turned over other things.
Me, I liked flipping over logs and looking for snakes. When I was about 11 or 12, I started keeping snakes. And my selection of what I wanted to have was entirely based on aesthetics. I really liked the color patterns.
Having spotted so many patterns as a child, Carroll has spent his entire career asking, how do such patterns form? How do you explain all of nature's diversity?
When I was a graduate student or beginning work in this field, biologists could look at protein sequences fairly well, but they really didn't understand anything about the networks of development. In fact, no genes involved in controlling development had been identified and isolated.
After earning his Ph.D. in 1983 from Tufts University, he did a postdoc at the University of Colorado. By that time, embryologists were starting to isolate developmental genes. For example, ones that determine front versus back or head versus tail. Carroll showed that a series of such genes could sculpt forms in an intricate dance of turning on and turning off other genes. Most surprisingly, these genes were found to be similar in creatures as diverse as worms, elephants, people, and flies.
The expectation was that we'd be seeing evolution at the protein level.
In 2005, Carroll pointed out,
there are two parts to genes. There's the functional part that encodes a protein that does the work in cells. But there's another part to every gene, and that's the part that contains the instructions for where that gene will be used.
Paradoxically, it turns out that there are too few protein-coding genes, and they are too similar across species to account for the tremendous variety of forms.
And that paradox is resolved to understand that diversity is not so much a matter of different genes as using the same genes in different ways.
Carroll set up his own lab in 1987 at the University of Wisconsin at Madison.
My lab is focused on understanding how genes get used in different ways to generate the diversity of form that we see.
He became the leader of the field now known as Evo-Devo.
Well, Evo-Devo is short for evolutionary developmental biology, which is understanding the evolution of form.
In a series of thought-provoking papers, lectures, and popular books, Carroll was able to dredge evolutionary history out of the deep recesses of our DNA.
I think in 2005 sort of marked an inflection where we were able to start pinpointing the exact DNA changes underlying the evolution of specific traits.
Changing when a particular gene is turned on can add spots or even change an antenna into a leg. These body-building systems form a special class of genes.
We refer to them as the genetic toolkit for development. And the analogy to a tool is this, is that, a carpenter with a particular tool can build just about any structure from the same starting materials.
The fruit of that theory is a new understanding of how evolution can create the endless forms most beautiful that first puzzled Darwin.
I think it's the second golden age in evolutionary biology, the first age being the age of exploration led by Darwin and Wallace and Bates and their contemporaries. But 150 years later, we're in a new golden age because we can pinpoint with exact precision the mechanistic causes of evolution.
Carroll divides his time between his lab and the Howard Hughes Medical Institute's Education Division. He continues to write books and make films for the general public. For Carroll, the scientist's life is one of adventure.
We're naturally explorers, and you start out as a little kid flipping over logs and seeing what's there, and maybe as a big kid you crack rocks and you find what fossils are inside, or you open up embryos and figure out how genes build that. And that's the adventure. Whether it's out in the jungle or whether it's inside the laboratory, it's exploring the world and understanding how we got here.
The 2012 Benjamin Franklin Medal in Life Science was presented to Sean Carroll for proposing and demonstrating that the diversity and multiplicity of animal life is largely due to the different ways that the same genes are regulated, rather than mutation of the genes themselves.